This is lecture 29. It's the second one on anabolic cellular signaling. So it's part two of seven on this topic. And we'll pick up where the last one left off. This was the slide we ended on, our checkpoint. So what is mTOR? It's a cell signaling cascade. mTOR itself it's an enzyme, right? It's a kinase. But mTOR signaling is the primary path your skeletal muscle takes to remodel itself. It's not just skeletal muscle. mTOR is the same path for anabolism that other cells take too, but downstream from mTOR, the stuff that gets phosphorylated is aimed at translation, which means protein synthesis. So the remaining questions, what is an mTOR complex? How many complexes are there? What does mTOR signaling do? How does it work and what does it accomplish? That's what we're going to be talking about today. In the last lecture, we started looking at the details of cell signaling, plump look at PI3K, PKB activity, and then more slender look at some of the other cascades. In this lecture, though, it's just mTOR. mTOR specifically, that's all we're looking at for the most part. And upstream and downstream from mTOR in that cascade, that's the stuff we're going to be looking at today. This is the actual cellular cause of hypertrophy and proliferation in a bunch of areas, a bunch of tissues, like in the brain too, like dendrite formation, proliferation, differentiation, migration. All of this stuff is regulated by mTOR. So throughout your whole body, inside of your cells, you're going to see mTOR relaying its signals. So let's talk about the physiology of those signals. The complicated regulatory government of your cells exists in protein complexes. And this just means proteins that travel together, little families, they have different members. There's a husband and wife and daughter and son, puppy and whatever. They all live together. But like nobody pretends, the puppy and the mom are like the same person. They just live and travel together. So that's what protein complexes are. It's proteins sort of in a household. And this article has a nice summary of mTOR complexes 1 and 2. And these are sort of the main proteins in each of those two complexes, depending on who you're asking. There are more than this, but these are the core proteins in mTOR complex 1 and complex 2. Complex 1 is what we're going to be focusing on. We'll talk about complex 2 today, but mainly from the perspective that it's an upstream activator of complex 1 and then the basics of its other signaling functions. But mTOR complex 1, this is the primary path, the primary conduit through which anabolic signals are delivered, is complex 1. So think of the core proteins in the complex, like baseball or softball players. If you want a functional system, like if you want a functional team, yeah, there are three reasonable positions to put on the field, right? A pitcher, a catcher, and some shallow outfielder. It's not a good defense, right? But it suffices for like a home run derby. If you only get three players, this is a reasonable construction of defense, the core hub. Um, and mTOR also has a core hub. You can think of the mTOR enzyme like the pitcher, but the pitcher isn't the whole team. Just like the mTOR enzyme isn't the whole complex, but it's critical for the game to be played. So in this case, mTOR is a kinase. It's what does the phosphorylating of downstream targets. And then the catcher here, we'll call that raptor, regulatory associated protein of mTOR, RAP-TOR, R-A-P-TOR. So mTOR has to pitch its phosphates somewhere specific, and Raptor provides its targets. I'll explain that in a minute. And then The last one here would be MLST8 or Gable, G beta L, G protein beta subunit like. So Gable or MLST8, two names for the same thing, in this case, a center fielder. Critical, right? Gable is, it's critical, but more peripheral and far less interesting than the pitcher and catcher. Again, we'll talk about it in a couple of minutes. But if you want to play the full game, if you want to play baseball or softball, there's a third baseman, right? Or base woman. There's some infielders, more outfielders. There's a batter, right? These things like Tubrin and Reb, they're definitely players on the field, right? Very important ones. They're just not as central as the pitcher and catcher. But here on the side of the screen, we have 10 players, right? There's 9 fielders and a batter. Um, and if you want the game to function, right, at the MLB level, you need like umps, right? You need coaches and base coaches, certainly more than one batter, right? You need like 9 of them. You need a whole opposing team, right? Maybe an athletic trainer, quadruple the playing field. So for mTOR to work, really what you need is a whole team of umpires, right? You need regulation. And without regulation, you die. So, like in baseball, if you don't have regulators wearing umpire jerseys, teams cheat, right? And then it's like people stop watching baseball. In life, in like regular human life, if you don't have regulators, banks misbehave, supplement manufacturers sell expensive placebos, if food becomes the cheapest source of poison you can buy, every financial market, finds itself like inflated to the point of implosion. Just ask Enron. And so we need to regulate everything. Everything needs to be regulated. Um, all function, I don't care what it is, impossible without regulation. Like even the regulators need to be regulated, right? Otherwise, you get corrupt administrative boards, and then like employees have to unionize and stuff. So it's everything is just regulation is so critical to function. And in biology, um, we need to regulate energy consumption, right? If there's an energy crisis, you die. So mTOR is tightly regulated, like snug shoelaces. If they come undone, it's a problem. Metabolism has double and triple knots. And there are some tricks that we can employ to sort of loosen the laces a little bit to favor catabolism, to favor, weight reduction, or to favor anabolism to grow and synthesize proteins. But we're not there yet. We'll get there in this lecture series. But first, we'll go through these proteins one at a time. We'll begin with mTOR, the enzyme itself. It's a kinase. It's encoded by the mTOR gene. And the protein has already had a few names. mTOR, the enzyme, it's had a few names already. It's unlikely that mTOR is the last name it's ever going to have. Um, this guy, Stuart Schreiber, called it FRAP. David Sabatini called it RAFT1. And then Robert Abraham is the guy who coined mTOR. And it stands for mammalian target of rapamycin. We'll talk about that name later, what rapamycin is. But that's the name that stuck. Sometimes people now will say mechanistic target of rapamycin, or they'll just drop the M and call it TOR. Someday, I assume we'll just drop the TOR altogether. Because the enzyme is so much more important than what TOR stands for. But again, TOR stands for target of rapamycin, which means is primary function, like literally what it's named for, is being the target of a chemical called rapamycin. And again, in a few minutes, I'll get to that. I'll talk about that a little bit later. Um, but just notice its existence on the left of the slide here, rapamycin. It's not myosin, it's mycin, and it's blocking the mTOR complex, which has mTOR, Raptor, and Gable in it. MLST8 or Gable here, it's listed as Gable G beta L. And then downstream from mTOR is a bunch of signaling for protein synthesis and cell proliferation. This is the main hub. Um, You can see MLST8 or Gable up at the top there, mTOR at the left, kind of all over the place, Raptor on the right. And this is a look at the protein structures in mTOR complex 1. But you'll notice this little guy in red up here, FKBP12. Um, this little FKBP12 is important. Okay, it's a binding protein that led to mTOR's discovery. In its original nomenclature, earlier I mentioned FRAP and RAFT1, these alternative names. Notice there's an F in both of those. Uh, F is for FKBP12, like FRAP and RAFTA, right? FKBP12 rapamycin associated protein, right? That's FRAP. Or rapamycin and FKBP12 target 1, that's RAFT1. Both of these used FKBP12 in their name. I mean, the eventual pen name of mTOR ignored FKBP12, but it's still referring to it. I mean, because like everyone knows what mTOR, what we're talking about, target of rapamycin. What's the actual target? FKBP12. So this little gal, let's say, appears critical to the discovery, um, as well as the science that followed of mTOR. So how do we identify what mTOR does? Block it, right? Block it with rapamycin and then figure out whatever biological systems go haywire. That pile of hay, right? That's what mTOR, the kinase, must have been like wiring or doing whatever. So if you block an enzyme, whatever the downstream consequences are, well, that enzyme was responsible for that stuff. So that FKBP12, the site of inhibition through rapamycin, is really important for understanding what mTOR does. Now, this here is Raptor, and you'll have to know more about Raptor than FKBP12. This is regulatory associated protein, RAP, of Raptor, regulatory associated protein of TOR. It's really abundant in skeletal muscle, less abundant, but still very present in other tissues, brain and guts and whatever, but really abundant in skeletal muscle. And Raptor is a major distinguishing characteristic of mTOR complex 1. Complexes 1 and 2 both have mTOR, obviously, and they both have MLST8 or Gable, but only complex 1 has Raptor. And Raptor has some diverse roles, but its primary role seems to be the recruitment of downstream targets for mTOR, 4EBP1 and P70S6K. So remember, mTOR is a kinase. It phosphorylates stuff. That's how mTOR sends its message. It attaches phosphates to things, but it can't just send phosphates out into the abyss and then hope they find their target. It needs a headhunter who can seek out candidates and bring them to mTOR. That's Raptor. Raptor is the recruiter for mTOR. So mTOR can phosphorylate stuff. And in times of nutrient surpluses or scarcity, Raptor is going to behave very differently. It gets turned on or off by phosphorylation, but it has multiple phosphorylation sites. So sometimes the attachment of a phosphate to Raptor can activate it. And then other times, depending on where that phosphate attaches, Raptor can be deactivated. Again, multiple phosphorylation sites. And then also the smallest bird right in the middle there, that's the actual Velociraptor, that like barely threatening Cretaceous turkey, right? That was like recast in the Jurassic Park series as like a terrifying genius. Yeah, it's that little tiny thing in the middle. Okay, so back to mTOR's Raptor. The directly upstream activator of mTOR is RHEB, R-H-E-B. And that has more diverse functions, including promoting Raptor, but we'll stay focused on its primary one, which is about the activation of mTOR, which we'll talk about soon. I just wanted to point out the relationship between Reb and Raptor. Again, sometimes phosphorylation can activate or promote Raptor, and then sometimes it deactivates or inhibits Raptor, depending on the site. With Reb activity, we see promotion, but other stuff inhibits Raptor. So Raptor, major site for mTOR interaction, lots of stuff phosphorylates it. Um, Again, some of that stuff promotes like MAPK, other stuff inhibits like AMPK, suspiciously similar names, but remarkably different kinases, MAPK and AMPK. We'll talk about both in detail, but for now, they both act on Raptor, among other sites. So don't think of Raptor phosphorylation as on or off. Just think of it as either. It just depends on the phosphorylation site, but you don't even have to memorize the sites. All you have to learn is the phosphorylating enzymes. Think of an enzyme as anabolic or catabolic. If it's an anabolic enzyme, it's turning on Raptor. If it's a catabolic enzyme, it's turning off Raptor. It's inhibiting Raptor. So this diagram is looking at RSK, which is an MAPK signaling separate cascade from mTOR. So here I'm just showing that these proteins have more than one function. Like, yes, RSK phosphorylates Raptor, but it has other interactions that turn on protein synthesis in both mTOR dependent ways and in mTOR independent ways. So you see the incoming arrow from ERK, extracellular signal regulated kinase, that's the MAPK cascade, ERK1 and 2, phosphorylate and activate RSK following the binding of MAPK stimulating hormones or growth factors or chemokines or whatever anabolic thing. Once you get RSK activated, it phosphorylates a lot of stuff that's aimed at anabolism and growth. Some of it is more mTOR crosstalk activity like Raptor, but it also has its own anabolic roadways as well. Um, Another form of mTOR crosstalk here is tuberin. RSK inhibits tuberin like PKB would. And you know that tuberin inhibits Reb, and Reb would be activating mTOR. So PKB and RSK inhibit the thing that suppresses mTOR activity, which obviously makes PKB and RSK very anabolic. And if you remember the Wnt proteins, WNT, the Wnt proteins, which inhibit GSK3, which would otherwise promote tuberin, this diagram should show some familiar territory. And then LKB1, which you see on the far right, that's something we'll talk about later in some detail, but it's an activator of AMPK, which is a major inhibitor of mTOR. RSK also suppresses BAD, right? We talked about that earlier, an apoptotic protein that PKB also suppresses. BLC2 associated agonist of cell death, so we're warding off apoptosis. Downstream from mTOR, RSK phosphorylates some of its targets, sort of unrelated to mTOR. It phosphorylates ATF4, so let's get some collagen. One of the major interactions of RSK that we care about here in this class and this lecture, crosstalk with mTOR. And we'll talk more and more about those. So just because I'm introducing a lot of names, a lot of enzyme names, it may feel overwhelming, but just bear with me and the repetition, a lot of this should start to set in. So it might feel overwhelming now, but this is only lecture 2 of 7. This will start to be more familiar. So in the last couple of slides, we were getting into MAPK, mitogen activated protein kinase, that signaling cascade, the one with RSK in it. Um, in the middle of these two pathways, right, mTOR and MAPK, there's crosstalk. The two major sites of crosstalk, of interaction, are tuberin and raptor. Um, those points where communication between these signaling cascades merge. And then at the end of the signaling cascades, they converge. They sort of land in the same place. And I'll talk more about that. But to sum up the Raptor stuff for now, Raptor is a critical protein in the mTOR hub that interacts with a lot of things, mostly the downstream targets of mTOR, P70S6K and 4EBP1. Raptor recruits for mTOR to phosphorylate. So mTOR can attach phosphates to those things. But Raptor is also sensitive to promoters like MAPK and inhibitors like AMPK. Okay, so moving on to MLST8 or Gable. Um, and if you've seen Interview with the Vampire, or read the book, but there's a movie. Maybe that'll help you remember this one. Probably not, but Lestat is Tom Cruise's character. But if we're talking about mTOR, right, so it'll be like M Lestat, right? MLST8. That, like, if you have a license plate of Tom Cruise's character Lestat, it would be like MLST8. Super corny. I apologize, but MLST8 is in both complexes, 1 and 2, and it's the least researched, it's the least understood, and it's the least important of the core proteins. When it gets deleted, mTOR complex 1 activity, right, the ongoing function of complex 1, is barely affected. So the role that MLST8 serves is more about assembly than activity. And it's much more important to complex 2's formation than complex 1. And if you get rid of MLST8, right, it doesn't really seem to affect downstream phosphorylation of 4EBP1, maybe a tiny bit of impairment to P70S6K, but it's not a pronounced effect. Um, and in some specific people, in some specific circumstances, MLST8 does have the potential to have important roles, like implications in cancer. If you knock out MLST8, you can reduce mTOR signaling in cancer cells. So it's not pointless. It's just not as pointy as some of the other proteins. And again, what seems to be its major role is assembly of mTOR complex 2. The MLST8 protein is sandwiched between the mTOR enzyme and mSyn1, which is a core protein in complex 2. We'll talk about that later in this lecture. So again, it's critical for the assembly of complex 2, but it's not particularly critical for ongoing function, especially complex 1. Okay, moving on to the group of proteins that are immediately upstream and downstream from mTOR. This diagram is really good. This is a useful diagram. So let's start with upstream. You see tuberous sclerosis complex, right? TSC1 and 2 are tuberin, right? Which is TSC2. But it's part of a tuberin, it's part of a complex. And this deactivates Reb. Tuberin deactivates Reb, which you see immediately upstream from mTOR complex 1. A major upstream input for mTOR, an upstream activator, is PI3K. So you go from PI3K to PKB to tuberin to Reb to mTOR. These are major landmarks on that road. And Reb is the actual point of connection here. Reb, RHEB, is what actually activates mTOR at the end of this PI3K highway. It's a GTP binding protein. So when when Reb has GTP and binds to mTOR, that activates it. Um, and the tuberous sclerosis protein complex functions as Reb's GTP ACE activating protein, a GAP, a GAP. So if tuberin, okay, let's say tuberin gets like mutated in a way that its function is compromised. Okay, you get tuberous sclerosis. Okay, tuberous sclerosis. That's a condition with out of control benign tumors, right? It's rare, but it's this overgrowth syndrome, and it's caused by a lack of mTOR regulation. Owing to that mutation of tuberous sclerosis complex. So that's why this little protein complex, that what tuberin is in, is the tuberous sclerosis complex. What it's doing, if it's working, if it's actually functioning, if you don't have a mutation, it's inhibiting Reb, and Reb is activating mTOR. GTP-loaded Reb is what actually turns on the kinase activity of the mTOR enzyme. And then downstream from mTOR, you see the stuff that mTOR, the kinase, is phosphorylating. Some of that stuff, right? S6K or S6K1 or P70S6K, all the same thing. That's all just different names for the same thing. Um, and that is a kinase. P70S6K is a kinase. And so mTOR kinases that kinase. mTOR attaches a phosphate to that kinase to turn it on. And then that kinase, P70S6K, phosphorylates RPS6. What you see is S6 there. That's ribosomal protein S6, RPS6. That's what really crosses the finish line, promoting synthesis of protein translation. And then 4EBP1, that does the opposite. That's an inhibitor of protein translation, and mTOR inhibits that inhibitor by phosphorylating it. Okay, this one isn't quite as clean as that last diagram, made it seem. But remember that mTOR is part of a complex with Raptor and Gable in it, or MLST8, Gable or MLST8. And the direct downstream targets of mTOR are 4EBP1 and P70S6K. Those have tasks of their own, right? When it's inactive, P70S6K is bound to EIF3. You don't need to know this, but when mTOR phosphorylates it, it detaches from that. And it's then free to phosphorylate its own targets. So RPS6, that's the only one you need to know. Ribosomal protein S6. That's what P70S6K is named after, P70 ribosomal protein S6 kinase. So P70 S6K phosphorylates RPS6. Okay, moving on from the immediate upstream and downstream proteins, there are a couple of core proteins, Deptor and Pras40, that are negative regulators of mTOR. And PKB and mTOR get these things phosphorylated, which relieves their inhibition. Deptor and mTOR. Deptor is interesting, way more than Pras40. So Deptor and mTOR, these are like quarreling siblings. They play this reciprocal regulation role. So if you ramp up mTOR's activity, you can reduce the expression of Deptor. But Deptor, in turn, is inhibiting the kinase activity of mTOR, so that mTOR doesn't phosphorylate its downstream targets very well. But like mTOR and Deptor, they're forever playing the role of sibling rivalries, but they each have their own lives. Right now, we know all about mTOR, but Deptor's private life, oh, that's a fascinating one. Because although it inhibits mTOR, it also inhibits apoptosis, right? It's not a one-dimensional character. mTOR is, right? mTOR is just grow, 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 divide, and grow, grow, grow some more, right? Hang out with mTOR and you know the story. You know what's going to happen. Deptor has deeper character development, right? It's an interesting protein. So it's both anti-catabolic and anti-anabolic. It's a morally gray character. So this is a graphic look at Deptor signaling. Yes, it inhibits the kinase activity of mTOR. Deptor does. Deptor inhibits the kinase activity of mTOR. So mTOR's downstream targets remain hungry for phosphates, but at the same time, Deptor is promoting PKB signaling and consequent proliferation, meaning more cells grow, then divide, and grow some more and divide. Um, so this is what I mean by Deptor is an interesting character. It's all like dark and brooding. It's like it's the mysterious little sibling of mTOR, less famous, hardly celebrated. It plays supporting roles, but man, it plays those roles compellingly. Deptor is interesting. But if you block mTOR, like Deptor does, you're blocking the thing that's inhibiting apoptosis and autophagy, the degradation and recycling of your cellular materials. And apoptosis is programmed cellular death. So when Deptor blocks mTOR, you're going to start shrinking, pretty quickly if you don't come up with a solution. Deptor finds its own solution. It creates the problem, but it solves it. So Deptor is fascinating. Again, Deptor and Pras40 are both negative regulators of mTOR. When they get phosphorylated, it reduces their interaction with mTOR, but they're not just simple stage props. Deptor and Pras40 are people too. Okay, so that gets us through the main players in mTOR signaling. The mTOR kinase itself, Raptor, which recruits mTOR's downstream target, so mTOR can phosphorylate them. MLST8, that assembly protein. The immediate upstream proteins, tuberin and Reb, and the immediate downstream proteins, P70S6K and 4EBP1. And then also RPS6, which is what P70S6K phosphorylates. So we have the sequence of proteins down. But mTOR, I talked about this a little bit earlier. mTOR, the name, mTOR, mammalian or mechanistic target of rapamycin. So it's named after rapamycin, right? So what is rapamycin? It's a big carbon compound, a macrolide. It's a big hunk of carbon produced by bacteria, and it was discovered on Easter Island, right? So it functions. Rapamycin functions as an exogenous, you know what that means, allosteric, you know what that means, inhibitor of mTOR complex 1. So our primary signaling system, balancing anabolism and catabolism in the body, is named after a chemical that doesn't exist in you that was discovered on a Polynesian island. There's like 1,000 islands off the coast of Chile. It's like a big triangle. And second only to those huge stone heads, rapamycin is the most famous thing to come out of that triangle. It's the foundation of metabolism. Well, rapamycin isn't because we don't actually make that. It's exogenous. But it led to the discovery of really the foundation of the balance between anabolism and catabolism. So at some point, mTOR's name, I suspect it'll change. But for now, mTOR, it has a nice ring to it. It sounds like an anabolic thing. And rapamycin is obviously used as a drug. Okay, it's a mTOR inhibitor. There's a ton of conditions, not just cancer, in which this drug can be of enormous value. Certainly not bodybuilding. I've taken it before anybody knew what my diagnosis was. They put me on everything. They put me on rapamycin. Um, but it's not just complex 1 that rapamycin is inhibiting. Um, some of the adverse effects of rapamycin et al., other drugs that are doing this too, come from complex 2 inhibition. It just takes time for that to happen because it's mostly the assembly of complex 2 that rapamycin is blocking, not the activity, not the kinase activity of complex 2, but the assembly of it. Whereas it blocks the kinase activity, the mTOR activity itself is seized in for complex 1. So this is a look at the two complexes. We haven't talked about RAG yet, and we will later, but everything else in the complex 1 chart: Deptor, Pras40, Raptor, MLST8, S6K1, P70S6K, and then 4EBP1. All of that stuff. The RAG GTPases, what you see over there, these are proteins that bind to mTOR, really to Raptor, when amino acids are abundant. It's a component of nutrient sensing. We'll talk all about it in a later lecture. And then on the right side, we have mTOR complex 2. And this has a lot of effects, broad metabolic effects: glucose uptake, ion transport, it regulates actin cytoskeleton organization. It has a lot of its own unique roles, totally distinct from complex 1. For the sake of this class, you don't need to know like any of them. Um, but you need to know some of the proteins on the complex 2 side. So obviously there's mTOR, MLST8, Deptor. You see those three, you know those three. There's also Richter, though, RICTOR, in the place of Raptor. Rapamycin insensitive companion of TOR, Richter. Um, there's mSyn1 and ProTOR over there. And the downstream targets are different too. One of them, well, PKB, AKT or PKB, same thing, two different names for the same thing. And that PKB is upstream from complex 1. So that means complex 2 is an activator of complex 1. But if you were to rely on complex 2 to do all of your complex 1 activating, you would be miserable, right? And puny, and then dead, right? It contributes to complex 1, but only as a part of a team of activators. It's a minor activator of complex 1. It would not be sufficient as a solo act. You need other stuff activating complex 1 also. So the other downstream targets of complex 2 are SGK, serum and glucocorticoid-induced protein kinase, and PKC, protein kinase C. PKC does a lot. It promotes the contraction of smooth muscle all over the place. Some other like digestive and kidney effects, tons of functions. We'll cover none of them. SGK, that regulates like epithelial sodium transport. It helps integrate some hormonal effects. Nothing you need to know. mSyn1, that's helpful in mTOR complex 2 formation, but it's also a recruiter for SGK, so mTOR can phosphorylate it. And then ProTOR is also involved in SGK phosphorylation, but you don't need to know the functions of these things, just the basics of complex 2. So looking a little bit closer at these functions, we'll just get through complex 2 in some detail, even though it's deeper detail than you'll be held accountable for. We've talked a fair amount about PKB, and a lot of its roles. We just finished barely covering SGK and PKC, ions and cell volume control, epithelial stuff, roles in liver function, cardiac function, beyond the scope of this class. The point of mentioning them at all is just to illustrate the metabolic breadth of these signaling cascades, in this case, the breadth of mTOR complex 2. A little bit more about SGK and mSyn1 falling outside of the hypertrophic channels, right, treading out into the wider world of metabolism. Again, ProTOR, another protein that helps recruit SGK and assist in the proper signaling of mTOR complex 2. In mouse models, if you knock out ProTOR, SGK phosphorylation is impaired. Okay, so let's link the two complexes though. Really, what you need to know about complex 2 is this: PKB, right? AKT or PKB. That's a downstream phosphorylation target of complex 2. And it's an upstream activator of complex 1 through its phosphorylation and deactivation of tubrin. But remember, if you rely on complex 2 to do all of your complex 1 activation, you are not going to be satisfied or alive. You need other things, other upstream promoters to contribute to PKB's activation, not just complex 2. And you can see what RHEB, Reb is doing in this diagram, sort of. So tuberin is breaking down Reb's GTP into GDP, guanine triphosphate into diphosphate. And you need GTP loaded Reb to bind to what's called the FAT domain of mTOR. That allosterically activates mTOR, as long as some other conditions are met too, which we'll talk about later. So when we zoom back out and look at signaling from this height, it does look like a big, ugly mess, but that mess should start to make at least some sense. You see the insulin receptor and its signaling, going through PI3K, PKB. You see tuberin and Reb. You see Pras40. Obviously, mTOR is in there. You see PKB interacting with AS160. So GLUT4 translocation can happen. PDE is in there, blocking PKA signaling to the right of mTOR. BAD and GSK3 and MAPK signaling, that's on the far right. Lots of things in here you should start to recognize. You should be starting to make a little bit of sense. And this slide, this just gives us the key on the left. So kinases and phosphatases and transcription factors and so on. You know that kinases transfer phosphate groups. They phosphorylate stuff, attach a phosphate to something. Phosphatases do the opposite. They remove phosphates from molecules. And so you can see what what kinases are in here, and what's up phosphatase, and you can see what's pro-apoptotic and pro-survival. The portrait starts to have some recognizable features if you take time, if you pause and take time to inspect it. Same diagram, right? But a new key. What do the lines and arrows mean? All those street signs that show how the traffic is navigating across the map. Again, the more you inspect it, the more sense it should make. But we'll go through this more and more in future lectures, and it should make more and more sense. So if we keep the same key, but focus on PKB specifically, we see how enormous of a hub PKB is. It just goes all over the place, its functions. So notice the types of downstream actions PKB has: survival, protein synthesis, glucose metabolism, and so on. So as we get to the end of this lecture, let's just do the narrative summary. Get rid of the diagrams and just put it into words. The story of hypertrophy. PI3K gets activated by something, insulin or some sort of growth factor, insulin-like growth factor or something like that. PI3K phosphorylates PIP2 into PIP3. Phosphatidylinositol bisphosphate becomes triphosphate. So PIP2 gets its third P, its third phosphate, and becomes PIP3, phosphatidylinositol triphosphate. We can actually stop the cascade here. We can reverse this with P10, PTEN, phosphatase intensin homolog, P10. This is a phosphatase. And as you know, phosphatases remove a phosphate. So P10, in this case, takes PIP3 back to PIP2. It strips a phosphate off of PIP3 and makes it PIP2. So we're not going to move forward to PKB. But if you do have PIP3, that PIP3 permits the activation of PKB by PDK, phosphoinositide dependent kinase 1, PDK. or you can, activate it with mTOR complex too, but you can only activate PKB once it's in position here. And that's what PIP3 is doing. It recruits PKB and PDK to the plasma membrane where PKB can be turned on. So now PKB is on, right? It's active. And active PKB has pretty much infinity effects, right? There's so many of them. But one of those effects, one major effect, is to phosphorylate tuberin, which shuts off tuberin. If tuberin were on, it would be converting Reb's GTP into GDP. And Reb Ross homolog enriched in brain. It has the word brain in its name, but it's ubiquitous, right? It's not just in your head, it's in your like quads and biceps, and it's everywhere. Um, so Reb GTP, when it accumulates, because TSC isn't breaking it down, tuberin isn't breaking it down, Reb binds to mTOR. Reb GTP binds to mTOR, which turns on mTOR's kinase activity. And then mTOR phosphorylates stuff. And the things it phosphorylates initiate translation. So we get an increase in translational capacity and efficiency. Efficiency is just a measurement of the protein per mRNA per hour. So just think of turning on translation, turning up protein translation. So the very, very, very general summary is this: stuff affects mTOR. Turns it on, turns it off. mTOR is tightly regulated. When mTOR is on, you experience translation. You experience skeletal muscle hypertrophy, the increase in cross-sectional area of those skeletal muscles. This happens by depositing more protein into those muscle cells. And where does this protein come from? Translation, right? How does translation occur? Mostly a bunch of mTOR signaling. Uh, Before we get to the review questions, I actually here, this one, um, this is from a David Sabatini article. He's one of the primary mTOR researchers. He's one of the founders of this branch of a study. This is one of his diagrams. It looks hectic, right? But once you start perusing your way through it, you see more and more familiar territory. Like historically, if you were to walk into my office, you see these like haphazard stacks of books and piles of paper. I mean, some of them half my height, accumulating in inconvenient places. It made my office like a little maze. But there was organization. To me, there was tons of organization. Like it looked mentally disturbed, right? But it worked perfectly. And then they remodeled the building and it destroyed my perfectly effective system of archiving. Um, but the same principle exists with mTOR. It looks like a huge mess, but it's far more effective and elegant than my office ever was. And the very elegant function of mTOR is homeostasis, right? Homeostasis of your metabolism. I mean, just like you can disrupt other components of your homeostasis, temperature, pH, blood gases, blood sugar, whatever, you can disrupt mTOR. You can disrupt your tightly regulated system of growth. And mTOR is an enormously important hub in the signaling cascades that balance anabolism and catabolism, the cellular signaling that attempts to maximize fitness through muscle metabolism, protein turnover, where this lecture series began. So if mTOR just throws in the towel, like, I give up, I can't do this shit, whatever, you'll catabolize yourself to death, right? And then opposite that, if mTOR gets overzealous for too long, you'll anabolize yourself to death. It's about balance. And all of those corresponding proteins, the Rebs and tuberins and AMPKs and raptors, all of this stuff, these teammates, these help mTOR achieve balance. But that balance can be tilted or tipped by behaviors and nutrition and supplements. And we'll get to all of that stuff. But in the next lecture, we'll talk about the specific inputs that mTOR is responding to positively and negatively, the variables that activate it and the ones that inhibit it. And eventually, before this lecture series ends, we'll get to the practical applications. Um, But again, before our review questions, I just want to show you some diagrams that my previous students have made. All of these are from memory. They weren't looking at anything. They just memorized the mTOR map, and this came out of their heads. It came out of a pen by way of their heads. And this is a whiteboard practice session before an exam, and it's pretty solid, a really clean diagram. It's a good one to look at and try to recognize things and to go back to this one as we move forward throughout this lecture series. To revert to look at this diagram again, you'll recognize more and more, and it'll make better and better sense. It's a really good one. And here's a nice, clean, tidy one. By the end of this lecture series, you'll recognize everything on here. This entire map will make perfect sense. This one is from Cynthia Villalobos. She got a really detailed in hers here. And if you want to follow through some of this stuff and just follow these roads along, you'll start recognizing more and more of these things. John O'Connor, this is from 2019, really tidy map in here. Now, students are drawing different things or highlighting different things, sometimes missing a different protein here and there, but these are all really good. Here's another pretty clean, easy to follow one. And I'm showing you all of these in case some of them make more sense to how your brain would map it. If you see a diagram that's like, oh, that's how I would draw it, then just pause and explore that one. And here's the last one we'll look at. Very tidy. It's missing a couple of things. Amino acid signaling is really limited here, but he got super detailed on the MAPK signaling. So strengths and weaknesses throughout these maps, but they're all good. All of these are good maps. And each one of these, the students produced without any notes at all. These are just the maps that they came up with in their own heads and memorized and drew. Most of those are on actual tests. I just scanned their tests, old tests. Okay, so here's the review questions for this lecture, and I will see you in the next one as we move one step closer to the end of anabolic cellular signaling.